Okay, welcome to our webinar today on Olympic Intertidal Ecology with Dr. Steve Fradkin. I'm going to introduce Rachel Brown in just a second here, but I just want to give you a couple of basic webinar housekeeping tips. So first of all, this session is being recorded. We'll be able to share the recording with you after the fact. We are also going to have the opportunity for questions today. Dr. Fradkin is going to take the questions at the end. So we're going to ask if you use the question and answer piece, the Q&A piece down at the bottom of your screen, and send in questions. We'll be offering those to Dr. Fradkin at the end of the talk. And now I'd like to introduce Rachel Brown, Faro's Education Manager, to get us started.
Hello, thank you, Melissa, and thank everyone for joining us this afternoon. I am Rachel Brown. I am Faro's Education Manager. Faro Marine Life Center is located in Port Angeles, Washington, and was founded in 1981 by a community leader and educator, Arthur D. Faro. We are committed to community education and specialize in local marine invertebrates and fish. Our presenter today is Steve Fradkin. Steve has been a coastal ecologist and limnologist at Olympic National Park since 2000. He is the chief scientist for the park's marine and lake resources. He conducts research and long-term monitoring on intertidal resources in Olympic, as well as San Juan Island National Historic Park and Lewis and Clark National Historic Park. He also conducts research and monitoring in the 650 mountain and large lowland lakes of Olympic National Park. He received his doctorate degree from Dartmouth College after earning his MS and BS at Michigan State University. Prior to coming to Olympic National Park, Steve conducted post-doctorate research on barnacle life history at University of Oregon's Institute of Marine Biology in Coos Bay, Oregon. It is my pleasure to introduce Steve Fradkin.
Thank you very much, Rachel, and thank you folks for showing up to this talk on such a wonderful day. It's a really nice day out there, so I really appreciate that you're taking your time to listen to me talk a little bit about some of our wonderful intertidal ecology that we have on the outer coast. So hopefully my slides are up now and you folks can see them. Yes, I am Steve Fradkin from Olympic National Park, and I've had the great pleasure of being able to work on the outer Washington coast for, this will be my 21st year. And it is truly a wonderful place. So what I'd like to do today is I'd like to talk a little bit about that place. I'd like to talk a little bit about some of the ecology of this really vibrant intertidal zone, that interface between the marine and the terrestrial. And I would like to talk a little bit about some of the research that and monitoring that I am conducting, that we're conducting in the Park Service to try to understand this, this really diverse and vibrant area a little bit more. So without further ado, I'll get into it. And by the way, thanks again to the Faro for asking me to give this talk. Always love talking about this stuff. Okay, most of you are probably familiar with where we are on the Olympic Peninsula, but just as a quick a place setting. The park has two units, this interior unit and then also a coastal strip, which is a 65-mile coastal strip that's about 1 to 3 miles wide on the outer coast of the Olympic Peninsula, facing the Pacific Ocean. It is the largest stretch of wilderness coastline, congressionally designated wilderness coastline in the lower 48 states. And one of the things to sort of keep in mind about this as a sense of place is that it is largely a roadless coast. The coasts of California and Oregon have Highway 101 that go right along them. 3/4 of our coastline does not have a road that goes right along it. So it really is a remote wilderness coastline, as this photo shows here. This is up around just off of Lake Cozette on the northern coast, around Cape Alava and Yellowbanks. And you can see that, that it is a wild and woolly coast, to be sure. One of the hallmarks of the Olympic coast is diversity. And one of the major diverse aspects of this coastline is a diversity of habitats. And I just love this, this Landsat photo of the Olympic Peninsula, because it really shows, I would argue, the value of a national park on the Olympic Peninsula, which was founded in the 1930s. And as a result, at the time that it was founded, you know, these lands were put into federal protection, and they, it was just a wilderness amongst wilderness. And now, from space, you can actually see the outline of Olympic National Park, both the coastal strip and the interior unit. You can see the untrammeled outlines of that as the lands around it have been developed for commercial forestry or for human habitation in the Puget Sound area in the North Olympic Peninsula. Okay, so this diversity of habitat types, this coastline really exists in a physical environment. And that physical environment is the Pacific Ocean. And the Pacific Ocean is a misnomer because there's nothing pacifying about it. It is a wave-swept environment. And the organisms that live here have evolved over millions of years to coexist and to survive that wave-swept environment. But it is still a harsh taskmaster, if you will. And it is a punishing environment. Here, this is just off of Rialto Beach. You can see James Island. And this is taken from this winter. We had many, many storms this winter of 25-foot plus seas. And these are beach logs that are being lifted off of the coastline and being tossed around in the surf. And one of the things that I'll talk about a little bit later is actually this storm season, this winter season, was really effective, more effective than any time in recent memory, of stripping a lot of this wood, this large woody debris from our coastline. And some of these trees are the size of buses. So this is a huge amount of wood. But onto the habitats. So we have, in this diversity of habitats, we've got rocky platforms, such as you see here, close to Chilean Memorial. And these are rocky benches that host rocky intertidal communities. We have here also another example of a rocky habitat is down in the Kalaloch area at Beach Trail 4, what we call Starfish Point. And this is a very different type of rocky area because this is a rocky outcrop surrounded by sort of a sea of sand. And that wave-borne sand really affects the type of biological communities that are able to persist on those rocky environments. So we get different communities that live on these sort of sand-dominated rocky areas as opposed to sort of rock-dominated areas. And I'll talk a little bit more about that in a bit. Then we also have boulder fields, these big, massive boulders that do not move around too much in the wave action, but in extreme events, they can move about. And so this is here just on the north, or on the south side of Beach Trail, of Third Beach at Taylor Point. And you can see these large boulders that are totally encrusted with macroalgae or seaweeds. And these are very diverse, rich environments. And they do have some disturbance that happens under extreme events, but by and large, communities are able to develop and to persist for a while with gaps forming every now and then. Then we have Cobble Beaches. This is down on the sort of the mid to southern coast around Mosquito Creek. And here you can see large cobbles the size of shoe boxes and things like that. And these move around a lot in the waves. And as you can see, there's not a whole lot growing on them because, as the old adage says, a rolling stone gathers no moss. And indeed, in the intertidal zone, rolling stones tend not to gather much macroalgae or marine invertebrates. But in some of the interstitial spaces in between, you can have organisms that do persist. So it's not a sterile environment by any stretch of the imagination. Then we also have gravel beaches, and this is Rialto Beach here. As you can see, the waves have sort of sorted the gravels out in a very aesthetically pleasing sort of way. But these gravel beaches are of a coarse enough consistency that there's not a lot that actually lives inside of those sand grains, because they're constantly being shifted around by the sand or by the waves, and things are ground up in there. And so there's not a lot that actually lives in these beaches, but this is actually a very important surf smelt spawning beach because they're particularly adapted to that type of environment. And then to sort of begin to round out some of our habitats here, we have sand beaches, fine sand beaches, and these tend to be on rather low angle, high surf beaches such as Kalaloch. And these beaches are fine enough sand that actually you do have fairly rich communities that actually live inside the sand, what we call infaunal communities, clams and a variety of crustaceans, amphipods and isopods and things like that. And I'll show you some of those a little bit later too. And by the way, in this picture, you see a bunch of people standing on the beach. That is my crew conducting razor clam population assessments, and I'll talk about that also in a little bit. Then, of course, not only do we have habitats on the mainland, but we have 490 odd offshore islands also that have intertidal habitats also, and these provide a different type of refuge from the mainland. And of course, the biggest offshore island that we have is Destruction Island, which is a particularly fascinating, relatively undisturbed area. And then we also have sea stacks, which are at low tide, are continuous with the land. And this is, you can see Hole in the Wall on Rialto Beach here, and some sea stacks right nearby. And then you put all these things together in the grand visage of Point of Arches on the northern shore. Just north of there is Shy Beach. But you have these long spines that go out into the Pacific that have sea caves in them that are just full of wonderful organisms that just blow my mind every time I'm there. I've been going there for over 20 years, and I am still amazed by the diversity in these areas. So I'm talking about the diversity of habitats. And of course, there are other aspects to these habitats because we get a lot of coastal driftwood, which forms habitat, not only for marine organisms, but also for terrestrial organisms that persist on the sidelines there. And as I mentioned a little bit earlier, that this year has been a particularly rough seas year where the ocean has stripped away a lot of this large woody debris. And this is a picture of Rialto Beach. And for those of you who are used to walking on Rialto Beach, this will probably look a little strange because where is all of the wood? That's a great question. I wish I knew the answer to it, but it's not on Rialto Beach. And there's also a lot of erosion that's happened. You can see some banks here that have very blunt faces because the sea has been carving away at the land in that battle between sea and land. And normally that large woody debris helps to serve as a buffer for some of that erosion. And coastal erosion not only happens that way, but we also get a ton of rain that falls on this rainforest coastline. And for a lot of our unconsolidated terrestrial sediments, they get waterlogged, and we get these mass wasting events like you have here on Third Beach. And that puts out a lot of sediment into organic debris into the intertidal zone, which helps to nourish beaches and also provides carbon into that coastal ecosystem. And of course, sometimes, since this is an eroding coastline, all those offshore islands used to be part of the mainland. And here we actually have a sea stack that several years ago that actually fell apart. So it is a constantly evolving coastline. The right-hand figure, you can see this sea stack right there, that's sort of high and dry, and about 80% of it fell off during a winter storm And it must have made one heck of a sound when that came down, because there were tons and tons of rock that came down there. So once again, not a pacifying environment. And also part of this habitat diversity story is, and this leads into the second diversity story, which is biological diversity. And one of the really cool things about the Olympic coast is we have all these diverse habitats that are jammed right next to each other. So this is up on the north shore of the park, right at the southern bit of Shy Beach, where Point of Arches comes in. And you can see that we have sea stacks, offshore islands, we've got sand beach, we've got cobble fields, we've got rocky platforms, all together in 1.5 kilometers. And you can even see in the waves here that there's a lot of sediment by the sandy beach. There is some sediment in this sand and gravelly area, and there's relatively little sediment in wave-borne sediment over by the more rock-dominated area. And of course, that has effects on the biological community structure of the organisms that live there. So one of the things about diverse biological assemblages is, or diverse communities, is that they tend to live in relatively diverse habitats that provide a variety of different niches for them, if you will. Okay, and of course, the intertidal zone of Olympic National Park is not an island unto itself. It is connected to that broader Pacific Ocean. And so that nearshore intertidal interchange of organisms and physical and chemical processes is very important. This is at Coastal Point here, just a little bit north of Hole in the Wall on Rialto Beach, and just a little bit south of Chilean Memorial. And you can see offshore kelp forests. That's what all those dark patches are. That's a really rich kelp forest there. Now, as I mentioned, there's that diversity of habitats. There's also that biological diversity. And this image here shows you just this rich assemblage of encrusting invertebrates and algae in the intertidal zone. And this is around Coastal Point on the mid coast. And one of the things that becomes apparent here is that because organisms have been evolving in this habitat in this environment for so long, is that real estate is at a premium here. They are constantly trying to find places to live and constantly battling with each other, both in predatory ways and in competitive ways for space. So it's a very tight real estate market in the intertidal zone. And as I mentioned, there's this nearshore and intertidal linkage. Almost all, or the great majority of the organisms that you find encrusting on rocks or embedded in the sand on our coast have complex life histories, is what we like to say, is that they have these life histories where they actually live in different environments at different life stages. And so the barnacle is an example of this. We're all reasonably familiar with an adult barnacle that lives stuck to a rock. But once they lay eggs, they actually have these larval stages that go out in the water column and go out into the nearshore ocean and are capable of moving tens to hundreds of kilometers for dispersal before finding a new home. So what happens in the nearshore environment is very important to what happens in the intertidal environment. It's good to keep those things in mind when thinking about coastal ecology. Now, biodiversity-wise, the Olympic coast is a biodiversity hotspot with arguably more species of marine invertebrates and macroalgae, or seaweeds, than any other place in the entire west coast of North America, from Alaska all the way down to Panama. We live in, it's a really, really rich area. We have over 200 species of seaweeds that we have documented. And in terms of fish, we have over 65 species of intertidal fish, some of which obligately always live in the intertidal, some of which are only in the intertidal when the waves, when it's inundated by the sea. And some of these fish, like the one in the upper left-hand corner, the snailfish, have these amazing adaptations. This is a pectoral fin, which is modified to a sucker. And you can sort of see the whole thing is teardrop shaped. It's basically a big mouth with a tail. This thing sucks itself onto rocks and just sits and waits for the tide to wash prey items over it before it can gulp it down. And then down in the lower right here, you can see that this is a juvenile rockfish. So the intertidal zone is an important nursery habitat for some of these commercially important species also. Okay, we have more than 530 species of marine invertebrates, from sea stars to nudibranchs and octopus and sea anemones and clams of all sorts, including razor clams. So an incredibly rich stew of organisms that calls the Olympic coast home. And a lot of these organisms have highly evolved behaviors. Here we have two beach hoppers or talitrid amphipods that are fighting each other for with sexual selection for a good mate. I refer to these things as the bull elk of the intertidal zone. As you can see here, my wife who works on elk has a picture of elk emulating talitrid amphipods. Okay, maybe that's not quite the right way, but that's the way I like to think about it. And then we also have a lot of birds that call the intertidal zone home. And so anyone who spent any time on our coast is probably familiar with the black oyster catcher, the loud, raucous black oyster catcher. And these are down in the Kalaloch zone here, feasting themselves on this surfeit of mussels. You'll also note that there are a lot of sea stars here, and I'll talk about sea stars in a bit. Then we have itinerant visitors to the intertidal zone. This is a coyote that has come down to forage. And so quite frequently we have mammals, including deer and even cougars that will come down to forage in the intertidal zone. And well, I suppose I have to put a nod to sea otters also, because they are pretty darn cute. We have a lot of sea otters on our coast. They were extirpated in the 1900s. They were reintroduced in the 1970s. And they have come back like gangbusters. In this lower figure here, you can see that they have just really increased. And this figure only goes to 2016, but we have, I believe, over 3,000 sea otters on our coast right now. And if you spend a little time on the coast looking for them, you probably will see them, particularly around rocky habitats. I want to talk now a little bit about sea stars and sea star diversity. And so, you know, we have a really diverse assemblage of sea stars on our coast. We have approximately 12 or so species of sea stars, described species of sea stars that run from leather stars to blood stars to sun stars to sunflower stars to bat stars. They are found in the high intertidal zone down to the very low intertidal zone. But there's one sea star that I want to sort of concentrate on for a little bit, which is Pisaster ocraceus, or the ochre sea star. And we have a very sea star rich and particularly Pisaster rich coastline. And these guys are voracious predators. They are the original keystone predator. And on our coast, they come in 3 different colors. What controls the colors? We really don't know. But they're all the same species. They come in this sort of brick red, this vibrant purple, and this orange color also. In other places like Puget Sound, not all 3 color morphs are there. But once again, we don't really understand what controls that. So science doesn't know everything quite yet. And you can see here in these other 2 images on the side, on the left-hand side, that these Pisaster are just slow motion marauding this village of mussels, their preferred prey. They are voracious predators. And down in this lower sort of humped island, you can see this band of sea stars foraging on the lower band of that mussel zone. And that leads us to this concept in intertidal ecology known as intertidal zonation, that if you look at the this upper right-hand figure, just from afar, you can see these different bands of organisms. And in general, these bands are set by a couple of different factors. The upper limits tend to be set by physiological tolerances. How much dryness or how much heat can a particular species bear? And the lower part of that bound is usually set by biological interactions, competition or predation. And in this regard, we're talking about predation. Here you can see in this left-hand figure, you can see all of these Pisaster ocraceus, these ochre sea stars, foraging and foraging and foraging, and they've wiped out all of the mussels below sort of a line that you could almost draw there. And that line is the comfort line for Pisaster ocraceus, for these sea stars. Above that, they get a little too dry and a little too hot. And so that elevational refuge, if you will, is what saves those mussels. If you take away those Pisaster, like the late great Bob Payne did from the University of Washington up on Tatoosh Island, did this foundational experiment in marine ecology where he took this keystone predator, that came up with the term. And a keystone predator is something that has an effect disproportionate to its abundance. So you don't need a whole lot of sea stars, even though the photo that I'm showing here has a lot of sea stars. You don't need a whole lot to have that effect. And he took a whole bunch of them away because they're relatively slow moving, and he watched those mussel bands go down in elevation. And then he put them back, and those mussel bands went back up in elevation. A very elegant foundational experiment in ecology. Well, Pisaster, by the way, means pea spine, because if you look at the very surface of a Pisaster, all these little glistening, wiggly things are the gills, and this big white thing is not a lesion. That is the madreporite, which is the opening to its vascular system, sort of its hydraulic system and how it moves. But these little white bumps here, I don't know why they look like peas to some taxonomists, but those are spines, and that's how this animal gets its Latin name. Well, in about 2013, not in about 2013, in 2013, there was a disease that was wrought on sea stars on our coast. And we have the dubious distinction at Olympic National Park to be the first place that sea star wasting disease was first observed. And that was in the summer of 2013. And relatively shortly thereafter, certainly within a year and a half, it was being observed all up and down the West Coast. And this wasting disease is just as it sounds. Sea stars basically started to lose arms and to begin to fall apart. It was really bad and really decimated populations. In certain areas in Puget Sound and in California, there was local extirpation, or all of the Pisaster died. And there's been a lot of work, some of which I've been a part of, that has been trying to find out what the cause is. And I'm sorry to say that we still don't know. We've had several false starts. At one point, we thought it was a type of adensovirus, but as it turns out, it's probably not adensovirus. And there's a recent paper that suggests that maybe it has to do with biofouling of the surface of sea stars. I take a little bit of a dim view of this because most of that work was done in the laboratory, and certainly in places like the Olympic coast, this wave-swept environment, our sea stars tend not to accumulate a lot of things on their surface that would stop oxygen from diffusing into them, and that is sort of what the purported main cause of this is. But at any rate, this wasting disease is manifested by lesions and falling off of arms, and there are a variety of potential insults that can do this. And it's still an active level of research to try to figure out what is going on with sea star wasting disease. But one of the good things is that the incidence of it has fallen off dramatically. And I'll talk a little bit about that. But first of all, one other thing about sea star wasting disease is that it is a remarkable marine disease event because for three reasons. The diversity of sea star species affected, over 20 different species have been affected by it. Its spatial extent from Alaska all the way down to Mexico, and also its temporal event, beginning in 2013 to the present day. Now, the present day, very low levels. About right now, I just did a survey about a week and a half ago, and it's at about 3.5% of the population, almost to the level that I'm not sure we would have noticed it if it hadn't been so bad before. But it's certainly still there now at low levels. Okay, so here's a figure showing at 4 different sites on the Olympic coast where the abundance of these sea stars at 4 different sites. And this red line here is 2013 when we first saw sea star wasting disease. And you can see that there was a sharp decline in certain populations. And actually, in all populations, there was a decline. We lost on average about 45 to 48% of our sea stars then. They have been coming back. Prior to that 2013 event, we rarely saw reproduction. And now we've seen about 5 different cohorts, 5 different instances of massive reproduction occurring. While I don't think that temperature causes sea star wasting disease, temperature certainly aggravates it. And this has been evident on our coast. Some of you may have heard of the blob, which was this anomalous warm water mass that was in the middle of that was in the Pacific Ocean in 2013, late 2013. It came ashore in late 2014, early 2015. And when it came ashore, we saw a big effect in water temperatures. And here you can see 2013. This is monthly water temperatures from 2005 to 2019. In 2013, you can see this spike in water temperatures here. And that's when we first saw sea star wasting disease. And then when the blob came ashore was in 2014, right around this area, this next peak, which is also a high spike. And also note this low spike here, which is the winter time. If you compare this winter to all the other winters, it was an anomalously warm winter time. So in each of these 3 instances, we saw spikes in sea star wasting disease. So this figure right here, while looking a little bit ugly, all you need to know is that the blue is a temperature anomaly. If it's going up a lot, like this here, here, and here, then that means it's anomalously warm. And the red bars are when we actually see a high proportion of sea star wasting disease. And so what you see, this is in 2013, during that first sort of warming event, when we saw sea star wasting disease. And then this is when the blob came ashore, where this big red circle is, and you see this big spike in sea star wasting disease. Almost 60% of the population was showing symptoms. And then we saw in that next winter, we saw sea star wasting disease also associated with warm waters. So what I would say is that sea star wasting disease is not caused by warm water necessarily, but warm water, anomalously warm water, certainly aggravates it and makes it far worse. And you know, as I mentioned, I've been talking a lot about Pisaster ocraceus, but Pisaster ocraceus, the ochre sea star, is not the only sea star that was affected. There's also a subtidal and very low intertidal sea star, the sunflower sea star, which is a very soft, very big sea star. They get to the size of almost manhole covers. So they're really big and they're voracious predators also. And we have not seen an adult sunflower sea star on the Olympic coast since 2013. In 2 years, we saw juveniles, but remember, these things spread in their larval stage, so they didn't necessarily come from the Olympic coast. Those larvae could have come from someplace else and then settled down on the Olympic coast. But certainly, we have not seen sunflower sea stars on the Olympic coast since 2013. And there's a move afoot to actually have the sunflower sea star declared a federally listed threatened species. Okay, I'm going to shift from one disease story to another one now, and I'm going to talk a little bit about razor clams. And razor clams are a very important ecological element on our sandy beaches, particularly on the south coast in the Kalaloch area. They're very important for nutrient recycling in the intertidal zone, between that linkage between intertidal and offshore and nearshore waters. And of course, they're also really tasty and a highly prized, sought after delicacy. Their range occurs all the way from Alaska, used to be down to California, but really the strong razor clam beaches really peter out in Oregon. And of course, here you can see people harvesting razor clams and what the razor clams used to look like at Kalaloch. Well, all right, maybe that's an exaggeration. And these are the types of signs that a razor clam will show in the sand, whether it be the classic donut or a little dimple. And this is how diggers sort of find them. They go around and look for those little holes, and then they dig in and they try not to break the shells, because they're called razor clams because the shells kind of look like straight razors if you squint, and they're also razor sharp if you break them. Okay, razor clams are particularly, to me, fascinating organisms. One of the reasons being is because they can dig like all get out. They have a very strong foot that they're able to push down in the sand and balloon the end like an anchor and then pull it in, thereby pulling the shell down, and then they just repeat that. And so they can dig sometimes faster than you can dig. And when you're digging for a razor clam, very frequently it is a race of diggers. So on Washington's shore, there are some very strong razor clam beaches, particularly to the south, Long Beach, Twin Harbors, Copalis, and Mock Rocks. The state beaches have very strong populations. And then probably the strongest northern population is the Kalaloch population within Olympic National Park. And that's what I'm going to talk a little bit about now. And the story here at Kalaloch is that the population has not been doing too well for the last decade or so. So in this figure, this shows the adult razor clam abundance in terms of millions of clams. And the different color codings are essentially whether it has been open to harvest or not. Purple are years where we've had open and free unfettered harvest. Green are where we've had to close harvest due to harmful algal blooms because they weren't safe to eat. And red is where we've closed them due to our concerns about the population. And so you can see in recent years, we've largely closed them due to concerns about the population. I would direct you to 2009, 2010, 2011, where you see this stepwise decrease, usually in the absence of any type of harvest, of about 50% of the population not being there the next year when we survey for them. And so, and in addition to that, not only have we seen these big drops in abundance, but the size structure we've seen huge changes in also. I'll just go to this slide right here, which shows you the average size of an adult. Now, an adult is considered a clam, any clam over 3 inches long. And what you can see since 1995 to the present day is there's been a steady decline in the size of an adult clam. And that is because adult clams are not surviving. We are seeing pretty good recruitment. These are the number of juveniles each year. And if I didn't have 2015 and 2017 in there, these others would look like reasonable recruitment. But these 2 years are just biblically phenomenal recruitment. Never seen before in any razor clam population to have 138 million baby clams recruit to a 4.1 mile beach. And that's the reason why you see in this slide here that we have these 2 big humps, because these are the survivors from the previous year. You can see in 2015, we had 138 million baby clams and less than 8 million survived to the next year. And then next year, very few of them survive. So clams the adult clams are dying off. Why is that? We think that there is a disease called Nix, which is a bacterium that infects the gill tissue. And this over onto the right-hand side, you can see up here, healthy gill tissue, and down below, relatively unhealthy gill tissue with these black dots being this bacterium, which goes inside of the gill cells and causes the nucleus to blow up and basically turns the gills to jelly. Gills are pretty important because they breathe through them and they die off. So here you can see in 2017, this massive die-off of clams. These are not clam shells. These are individual clams that are just flaccid and dying and lame and just being washed. They can't even keep themselves in the sand, digging in the sand, and all of these died. And so we've seen these massive die-offs occurring at Kalaloch. And we really think that it has to do this Nix bacterium, which, by the way, was responsible for a coastwide, all of Washington State, 95% of the razor clam populations died in the mid 1980s due to this pathogen. And we know that it's at Kalaloch already right now, and we've done some work with some USGS collaborators, US Geological Survey collaborators, where we developed a molecular probe, because the old way was using histology, which is very laborious. But we've developed a molecular probe, a modern technique, to see whether clams have this bacterium in them. And we took clams from Kalaloch, we took clams from one of the state beaches, Long Beach, down in the southern part of the state. And then we also took clams way up in Alaska, 2 sites in Alaska. And we have a paper that we published earlier this year on this. And one of the things that we saw is that in the Washington populations, 100% of the clams were infected by this bacterium. None of the Alaskan clams were affected by this bacterium. But the state beach to the south is not experiencing the population declines that Kalaloch is. And if anyone is actually a razor clammer here, you know that the state beach has been doing pretty well these last several years, where Kalaloch has not. And one hint at that is if you look at the number of copies of this genetic sequence that this DNA method identified, there is an order of magnitude more Nix DNA in Kalaloch clams than in the southern Washington state beach clams. So the level of infection for reasons as yet unknown at Kalaloch are way worse at Kalaloch. Not only are they all infected, but they're much more heavily infected. And we think that that is one of the major determinants of the reason why the population is not doing well. Okay, I've been talking a lot, and so I'll talk a little bit more. I've got a couple more topics to go through, but and I might not get to them, but I'll talk a little bit about our rocky intertidal monitoring program right now. Switching gears from disease ecology totally to tell you about some of the community structure that we have on our rocky intertidal shorelines. And so we have a long-term monitoring program at Olympic National Park where we look at the community structure of all of the invertebrates and the seaweeds that live on our coast. And we have, at particular sites, we have these replicate plots, and you can see these plots laid out in 2 different habitat types, the barnacle habitat and the mussel habitat. You can see some of the barnacle habitat over there and the mussel habitat in the foreground. And this is out on just north of Rialto Beach, is one of our index sites. And we just published a paper that's actually in press right now with a variety of other national parks on the West Coast and other sites too, because we're part of a consortium called Marine, the Multi-Agency Rocky Intertidal Network. And so there are over 100 sites along the West Coast where the same methodology is being done. And one of the take-home messages from this is that this middle graph here has these 4 lines, which are 4 different zones: the North Zone, the Central Zone, the Southern Zone, and then the island, the Channel Islands zone. And one of the things, and what we're seeing across years here, this is an index of community stability. I don't have the time to get into how we derive that, but you'll have to trust me or read the paper to ascertain that that is a reasonable index of community stability. And one of the things that we see is that over time, community stability is going down, but those southern populations really get rocked. And where they really get rocked are a couple of events, which are these warm water events, which really shift the community structure. And they shift that community structure to a more depauperate or a more depleted community. And our northern community is probably the most stable. And one of the reasons, as it turns out from this analysis, is because we have dominant and healthy mussel beds, which are foundational species that provide a lot of habitat for other species. And this other figure down here, what you can see, this upper left is the north, then the central, and the south, and then the Channel Islands. And this is the percent area cover, average percent area cover of mussels in the mussel zone. And you can see that in our area, the north, that it is relatively stable, if not increasing slightly. Not statistically significant, but relatively stable over this time period from 2005 to 2017. And in the south, you see that it is steadily decreasing, both in the Channel Islands and in Southern California. And a lot of those decreases really co-occur with these marine heat wave events. Okay, now we get affected by these marine heat wave events. And I already showed you with sea star wasting disease that marine heat wave events also impact disease ecology. All right, I'm at 45, but I'm just gonna plow ahead because I've just got one more set of slides here to talk about, and that's basically about ocean acidification. We are also heavily invested in monitoring ocean acidification on the outer coast. Ocean acidification essentially is that human beings have been burning a lot of fossil fuels, putting a lot of CO2 into the atmosphere. That CO2 likes to come into, that atmospheric CO2 likes to come into equilibrium with the ocean. And so CO2 dissolves into the ocean, forms carbonic acid, a weak acid, which then causes seawater, which is basic, to become a little less basic. All right. And and this was elegantly shown, and there's a great time series, which is this other figure here from Hawaii, showing a rise in CO2, which is the red, and that's concurrent with atmospheric deposition of CO2 by humans, and then a rise in seawater CO2, which is the green, and then a decline in pH. Okay, and so we have a site here in the mid-coast, the Focal Point, where we have some instrumentation in a tide pool that when it's inundated, as you can see in the lower right, is basically taking measurements on the water that all of these rich biological communities that are very highly susceptible to ocean acidification are being bathed in this water. So we're measuring that water that's affecting these organisms. And there is something called the saturation state of calcium carbonate, which pH affects. And I don't have the time to go into gory detail on it, unfortunately, but I would just say this, that this saturation state it basically is how soluble is calcium carbonate, which is the building block of animal shells, of clam shells and things like that. How soluble is that in the water? So basically at a saturation state of 1, below that, water is really corrosive, and that water tries to steal calcium carbonate from structures. Above that, there is somewhat ample calcium carbonate in the water. But in reality, anything below a 1.5 for, say, a clam or a mussel has a very hard time taking little calcium carbonate, or aragonite, which is the fancy word for it, taking those particles out of the water column to build the shell. And so anything below a 1.5 makes it really difficult for larval organisms to build that first home, if you will. And so these are data from Focal Point on our coast. And one of the disturbing things that you can see here is that we spend a lot of time over the course of years at a saturation state below 1.5. And we spend some time in corrosive waters where that water is actively eroding shells. Okay, so I've reached about my time. Of course, any of these topics I could have gone into in much greater detail, and I'm always happy to talk about these things with people. I have to acknowledge my collaborators and people who have funded me, and there's a grand smorgasbord of things here. Some of these are collaborators, and some of them are funders. Some of them are funders and collaborators. But at this point, I would like to stop jawing on at you on this beautiful day and leave you with this scene of Rialto Beach, and want to know whether there are any questions that I can ask. So yeah, thank you very much for your attention.
Thank you so much, Steve. We do have several questions in our question box. I'm gonna just kind of try to group them so they're a little easier to organize for you. And I'm gonna go backwards first. So there's a question about the blob. And the questioner wants to know if the blob could contain more than just warm temperature. So it could be hosting different bacteria or chemicals that are normally found at the coast that may have been picked up or from elsewhere in the Pacific. And could that be also influencing the marine life?
Well, we don't have data to suggest that that's the case, but the thing about the blob, how the blob formed was not necessarily that some warm water came in and parked there, but rather the water that was warmed by the summer before, warmed the water, and then that water never adequately cooled down that next winter. And so that warm water persisted. Now, of course, water, that is warmer has a different density than water that is colder. So, and you can do this experiment in your bathtub. And if you've ever swum in Lake Crescent during the summer, you know this. You're swimming at the surface and there's that warm water lens. You go down a couple feet, whoa, it's cold. And so that blob out there, while that's sort of natural water out there, that does become a bit isolated from the rest of the ocean because it's not mixing with the ocean. So you could have bacteria or other plankton proliferate in the blob, and then as it's blowing ashore, you could have that then being inundating the organisms on shore. But what I would add is when we first saw sea star wasting disease, it wasn't the blob that we saw. That wasn't the first time warm water hit us. We actually saw warm water in 2013, which was pre-blob. And unfortunately, climate change and global warming is leading to an increase in marine heat wave events. And unfortunately, these sorts of warm water anomalies are going to become less anomalous as time goes by.
So thanks, Steve. Related to that, someone wants to know if the sea star wasting virus, which we've decided now is probably not a virus, correct? But if the sea star wasting disease has affected any other organisms besides sea stars that we know of.
Yeah, that's a great question. And the answer is, unfortunately, we don't know. Back when we thought it was a densovirus, which is a type of virus, there was a line of inquiry to look in other organisms to see, and certainly also now post-sea star wasting disease, the marine community writ large is actively looking for signs of disease elsewhere. Sea star wasting disease is the largest, most significant marine disease event ever recorded on Earth. And that's not hyperbole, that's just a fact. And so it has really clued people into looking at other types of organisms. And certainly marine diseases are not new under the sun. But what we're looking for with marine diseases is sort of an increased frequency and magnitude of those things. And unfortunately, with sea star wasting disease, since we don't know truly what the cause is, we actually don't know that it truly is a pathogen like a bacterium or a virus. It might be, but we actually don't have compelling evidence for that right now. I mean, if it was, back when we thought it was a densovirus, there are a whole class of densoviruses that could potentially infect other organisms. We like to think that science pretty much knows everything. Unfortunately, and we know a lot about big stuff, but it's the small stuff that science is just now beginning to crack the lid on, and particular microbial ecology. A milliliter of seawater has hundreds of thousands of bacterial and viral particles, which by and large are almost wholly undescribed by science. So we have very crude tools still to understand these things, and we've got crude understanding of them. So I definitely wouldn't want to rule out that there is some sort of biological pathogen associated with it. But a lot of the quick fixes, that it is some sort of a known toxin, or that it is just temperature, or that it is, an easy virus or bacterium. We've largely excluded a lot of those things, but I wouldn't want to pretend that we know what the cause is yet. So it's an unsatisfying answer.
Never turns out to be as easy as we want it to be, for sure. Okay, so we don't have too much time left, so I'm going to move on to some questions that are a little easier maybe to answer. So first of all, someone is curious if the sediments released by the Elwha River dam removal have reached beaches on the outer coast.
Yeah, I don't think so, I don't think in a significant way, and that basically has to do with how those sediment plumes occur. Now, of course, sediment is not a monolithic thing. Sediment, is a gradient of really coarse material and really fine material, and that fine material stays aloft in the water column much longer, whereas that coarse sediment really drops out a lot and gets spread more locally and builds and nourishes local beaches and maybe even a little bit of Edith's Hook, but most of Edith's Hook is nourished by bluff erosion. So if there is sediment that has made it to the outer coast, it's probably very fine stuff, and it's not a whole lot that's made it out there. Because remember, water is sloshing back and forth into the Salish Sea and out of the Salish Sea. And there's ample opportunity for that sediment to settle out well before it rounds the horn and gets to the outer coast.
Great.
The offshore, or the rivers, the major rivers on the outer coast are much bigger sediment inputs.
Oh, great point about that. So we have someone who's interested in learning if you could recommend technically a beach for high seaweed and invertebrate diversity for someone who has very little experience on the outer coast. So where would you send them? Well,
Beach Trail 4, the Starfish Point area, is a really good place to look at that. There's really easy access there. Of course, if you want to have your mind boggled by diversity, go out to Cape Lava during a low tide and start moving some cobbles and rocks. Because as I mentioned, cobble is, in really high wave areas, tends not to be terribly rich, but in areas that are relatively protected, like Cape Lava is by nearshore kelp force and some of those islands there, it's really diverse and rich. And so I would recommend Cape Lava and also Beach Trail 4, Starfish Point.
Great, thank you. Okay, just one quick, really this is more of a general combination of several questions about tide pooling specifically. The question has to do with whether or not people physically out there in the tide pools can harm animals such as barnacles and mussels by walking on them, and whether that walking then contributes to potentially lower diversity in any particular tide pool region.
Well, the answer is yes, and kind of. So to go a little bit more in detail, certainly walking on organisms has the potential to harm them. Some organisms are more robust and are able to deal with it than others. And it also depends how you walk on them. If you wear Vibram-soled hiking boots, you're going to crush a lot of things. If you wear extra tough boots, which have a much more softer sole, and you tread lightly, then you do a lot less damage. A long time ago, an employee and a student of mine, Alita Erickson, did her master's degree on intertidal trampling, where we looked at a variety of sites along the outer coast and paired them for easy access and hard access at each site. And now some areas on the coast are really easy to access, like Hole in the Wall and Beach Trail 4, and get a lot of visitation. other areas are really remote. And the upshot of that research effort was that by and large, we don't have an intertidal trampling problem on our outer coast, except for at a couple of sort of sacrifice zones, which are these two areas that I just mentioned, right at Hole in the Wall and also at Beach Trail 4. But those impacts are also highly localized. You can see them, and barnacles will actually adapt their growth form to be more squat and more robust down on the rock in the face of trampling. So these animals have incredible adaptations. But without a doubt, trampling, particularly frequent trampling, hurts organisms and can affect community structure. This has been particularly seen at other areas. Now, the Olympic coast, we have these sort of high impact areas that I just mentioned, but you go down to, say, Cabrillo National Monument down in San Diego, which gets like 2 orders of magnitude more visitation than our areas get. And their trampling is a huge issue. So, you know, with time, we're certainly seeing increases in park visitation, and particularly this past year, but it's been increasing even pre-COVID times. And we might get to a point where we do begin to see more impacts on the population level. Certainly on the individual level, there are impacts.
Great, thank you so much, Steve. This is the technically the 5 o'clock hour. I think Rachel might have one more slide with contact information on it if you're willing to have people send you any additional questions that they might have.
Absolutely. As you can probably tell, I like talking about this stuff. And if I've not been clear or I've piqued your interest in a way, do not hesitate to contact me, and I'll try my best to give you a cogent, timely answer.
All right, so Rachel's just about to pop that up on the screen, but I will just reiterate that we have recorded this presentation and we'll be making it available on our YouTube channel. So you can see there we've got Steve's email and also Rachel's email if you have any other questions. We do have one more program coming up in our speaker series. That's going to be Tuesday, May 18th, again at 4 o'clock, and we'll be discussing more about that Elwha River restoration project and the dam removal with Jamie Michelle from the Coastal Watershed Institute. So I imagine all the questions we got around Elwha River will be further answered at that time. So yeah, I want to thank you all for attending, and thanks, Steve, for presenting.
Hey, thank you.
I always learn a ton when we have you, so I appreciate you to no end.
Well, thanks, Paul, and thanks to the Pharaoh. What a great organization.
Well, thank you. All right, well, everyone, hope you have a great evening, and we will see you again soon. Take care.